I've worked on a mouse embryo for all my research career. I can still look down a microscope and see that embryo, which is the size of a speck of dust, just a few cells. It's starting to make a cavity. It's the beginning of life. And to see that, to watch it, to understand how that is genetically controlled in more and more exquisite detail, to me is the most satisfying thing. We've worked on the mouse embryo for many, many years, trying to understand how an egg becomes a specialized human being or a mouse, and that involves cells becoming more and more specialized as development goes on. My lab at the moment is interested in, can we drive these pluripotent cells to become endoderm cells that would make the gut, would make liver, would make lung, would make pancreas. And we've been doing that by overexpressing particular genes that we know are important for that particular cell type. So we've been looking at how the cells make decisions in the embryo and trying to use that information to get stem cells to be able to make the right decisions, to make cell types that could be useful one day for therapy. Embryonic stem cells come from what's called the blastocysts, the pluripotent cells in the mouse that give rise to the whole mouse. But at the same time, we've been studying and know that there are other cell types that form in the early embryo that make the placenta and make other membranes that a baby needs to survive in the uterus. So we'd always been wondering whether there are other stem cells you could get from this same stage. So we were able to show that you could derive trophoblast stem cells that also replicate indefinitely in culture. But now they don't make pluripotent cells, they don't make the cells of the body, but they make the cells of the placenta. So in the mouse, we can actually use them to study how the placenta forms. We're working very hard to try to get human trophoblast stem cells to be able to understand some of the problems in human pregnancy with regard to placental insufficiency, preeclampsia. A lot of pregnancy problems come from problems in the placenta. Stem cell research is very important for cell-based therapy. That's really what the public excitement is. And there are lots of diseases that one could envisage stem cell treatments having a role in. Diabetes, Parkinson's disease, spinal cord injury, a variety of diseases. But there's other exciting things about stem cells too. They're not only cells that you can use to treat disease, but they're cells we can use to help understand disease and maybe develop other kinds of therapies, drug therapies and so on. We're going to see a huge growth in the use of stem cells for testing drug toxicity, but also developing new drug screens. And big pharma, small pharma, biotechs, labs, many people are excited by this potential. So I think we're going to see their use as a tool become much more widely applicable in a wide range of clinical and commercial settings in the next 2 or 3 years. The whole concept of developing drugs in the sort of pharmaceutical industry has always been on the idea of one big blockbuster drug that will treat everything, because that's how you're going to make money. What we know, of course, is that human beings are all different. So the whole concept of one drug that will treat everybody is going to have to change. And it's changing with really the concept of sort of personalized medicine. Stem cells have an impact in here too. A lot of that is focused on really genomics. Let's look at genetic variation between people, but we need to understand the function of those different genetic differences. If we can make stem cells from people with these different genetic variants, and then we can make heart cells and see whether this particular variant affects how a drug treats the cell. So there's a whole load of ways that we can combine personalized drug therapy with personalized stem cell medicine as well. We have excellent facilities here. In Canada in general, I would say our infrastructure facilities and our equipment and platform technologies are really at the cutting edge in every area you might want to be. And a lot of that has come about because of the investment through the Canada Foundation for Innovation. which has brought federal money and provincial money through the Ministry of Research and Innovation in a match to really ensure that in all areas of research, not just health research, we really have the best facilities available. We've established here the Ontario-induced pluripotent stem cell facility, where we're generating some of these new reprogrammed cells from clinical disease. The Ontario Institute for Cancer Research, which is a major new initiative in cancer research here in Ontario, has several different themes, but one of them is cancer stem cells. I'm a co-PI on a project called NORCOM, which is a Genome Canada-funded initiative jointly out of Toronto, Winnipeg, Calgary, to contribute to the International Knockout Mouse Consortium. We also have excellent access to mouse models of disease through the setup of the Toronto Centre for Phenogenomics, which is an interinstitutional facility. So right here in the sort of heart of the Discovery District in Toronto, we have the University of Toronto and several of the teaching hospitals, all in close proximity. We can walk from place to place. And in fact, in the building that I'm sitting in here, we have some of the best stem cell researchers all in one building. Not all in one institute, both in Sick Kids and in the University Health Network. We're all in one building. We can meet, we can interact. Also, because we have hospital environments, we have great interactions with clinician scientists to provide us with materials to isolate new stem cells and, of course, in the end, to start working towards therapeutic applications. So it's a real convergence of the basic science at different interdisciplinary levels and the ability to take it through to the clinic. But of course, to make stem cell research really strong, you need other kinds of science too. So the whole Discovery District is incredibly strong also in proteomics, in genomics, many of the bioimaging, many of the platform technologies that also are needed to help us move forward. So it's a really great collaborative environment. Stem cell research is an incredibly fast-moving field. It's very hard to keep up. One of the ways that we're very good in Toronto, in Ontario, and in Canada in keeping up is that we work together. What we have that other centres in the United States don't have is this real culture of collaboration. That there really is a lot of interaction. We have what's called the Toronto Stem Cell Initiative. All the stem cell groups are on a website together. We have monthly meetings. We have grants that we hold together, a number of new grants linking those different institutions, different researchers. So there's a real history and a drive to work together and collaborate. So platform technologies that one institution holds are available to all.